Hi, this is Lacey Havely. I will be doing an educational presentation on epigenetics for an AP biology course. This is for my Biology 510, a comprehensive cell and molecular biology course at Grand Canyon University. Epigenetics has a couple of objectives for an AP biology course, including explaining the science of epigenetics, being able to describe how epigenetic modifications lead to changes in gene expression, being able to describe the various modification types of epigenetics that occur in both DNA and chromatin, and then also identify and describe positive and negative effects of epigenetic changes. So what is epigenetics? Well, this is a process that alters gene activity without changing the DNA sequence. So this means that it's turning genes on or off and determining which proteins are going to be transcribed. So these changes that occur may lead to changes that can be transmitted to daughter cells. So through the process of mitosis and meiosis, these daughter cells that are produced are going to have these changes in their DNA. These may actually occur naturally. So when we think of a change in DNA, we immediately think of something negative like a mutation, but that's not necessarily true. These are things that are needed through our natural development for normal functioning, but in some cases may lead to health and behavioral effects. This has been linked, epigenetic changes have been linked to almost all cancer types, cognitive dysfunctions, and various other illnesses in respiratory illnesses, cardiovascular, reproductive, autoimmune, neurobehavioral. So there can definitely be some negative things that come with changes in epigenetics. There are three types of modifications: DNA methylation, histone modification, and non-coding RNA. DNA methylation is when methyl groups attach to DNA buildings. This means that a gene is turned off or silenced and no protein is produced. It does play a role in cell differentiation and embryonic development. So when we have our sperm and egg cells joined together to create a life, there's only these two cells. So how do we get from having only two cells to the trillions of cells that we do now? That's through cell division, but then also through cell differentiation, which is a part of DNA methylation's job. So methylation turns genes off, whereas demethylation turns genes on. And so this has actually been linked to certain cancers, lupus, muscular dystrophy, and a variety of birth defects. It shouldn't be surprising though, because when we think about DNA methylation, like I said previously, it does play a role in embryonic development. And so it's not surprising that it has been linked to certain birth defects. The second type, histone modification, well, histones are proteins that make up chromatin. And they are modified by the addition or removal of chemical groups, usually acetylation and methylation. And so chemical groups determine how tightly the DNA is wrapped around its histone, which affects genes being turned on or off. And so this is involved in the manipulation and expression of DNA. So when modified, they are translated into a protein, which influences how chromatin is arranged and determines whether the chromosomal DNA is going to be transcribed. So if it is a non-compact chromatin, it means it is actually active and DNA can be transcribed. However, if it is a compact chromatin, this means that it is inactive and DNA cannot be transcribed. This can lead to abnormal gene activity or abnormal gene inactivity and has been linked to many common genetic disorders. Finally, non-coding RNA controls gene expression by attaching to coding RNA to break it down so that it can no longer make proteins. It can also gather proteins to modify histones so that they may turn genes on and or off. This type of non-coding RNA does play a major role in brain development and function. So there it is, occurring naturally. Now, a non-natural or an epigenetic change has been linked to certain psychiatric disorders. Again, not that surprising though, because if non-coding RNA plays a major role in brain development and function, then it's not that surprising that it has been linked to certain psychiatric disorders. Things like schizophrenia, bipolar disorder, and major depressive disorder have all been linked to this non-coding RNA. So how can epigenetics change? Well, there's 4 different ways: development, age, reversibility, and health. What do I mean when I talk about development? Well, we kind of already discussed that a little bit already. So we have only the sperm and egg cell, yet when we are born, we have hundreds, thousands, millions of others, different types of cells. So how do we get all of those? Well, it's that differentiation. So they determine which function a cell will have. Will it be a brain cell? Will it be a heart cell? Will it be a liver cell, a stomach cell, a skin cell? But it allows cells to turn on and make certain proteins that are needed for its job and turn off the genes that are not important for the job. So if we're looking at a brain cell, it's going to have the genes turned on that it needs to help it function as a brain cell, and it's going to have the genes turned off to help it function as a stomach cell. Right? And that's how we get that differentiation of cells. So age. So as we know, cells change as you get older. So the level of DNA methylation actually does decrease over time. So a newborn is going to have the highest level of DNA methylation, whereas someone that's like 89, 90, 100, they're going to have a lot less DNA methylation in their cells. A 32-year-old is going to be somewhere in the middle. And this is because these cells have not divided nearly as often as they have when they are brand new. So these cells are going to have a lot more DNA methylation because of a newborn, because they haven't divided nearly as much. By the time you get to being 89, 90, 100 years old, you're going to have those cells are going to have divided quite more often and be the daughter of a lot older cell and have some of those changes already to it. But some of these changes are reversible. So they're not necessarily permanent and may be added or removed in response to certain changes in the environment or in the organism's behavior. So a really great example of this is smoking. So smokers tend to have less DNA methylation than non-smokers, but people who were once former smokers tend to see an increase in their DNA methylation as early as a year after quitting, getting it basically right at, or if not the same as somebody who was a non-smoker. So it can be reversible. And then finally, health. So infections weaken our immune system, which helps genes survive. A really good example of this is tuberculosis, TB, which actually causes a histone change that turns off the specific gene IL-12B, which weakens the immune system. Cancer, so just like certain mutations can make you more likely to develop a cancer, so can epigenetic changes. So for instance, an increase in DNA methylation, which decreases the amount of BRCA1 gene, so an increase in DNA methylation, decrease of BRCA1 gene, makes a person a higher risk for getting breast cancer than the other cancers. However, having this increase in DNA methylation does help to determine which cancer a person may have and even possibly detect the cancer sooner. So there is some possible benefits to this study of this topic. And then, of course, nutrition during pregnancy, we know, is super, super important for not only the health of the mother, but also the health of the baby. But the behavior and environment that a mother has during a pregnancy can change the epigenetics of the baby. And these changes may be short-term, but some of them can be lifelong changes where they may be more susceptible to certain diseases. So for instance, if a mother is pregnant during a famine, their child is more susceptible to develop diseases like type 2 diabetes, heart disease, schizophrenia, and these are all due to the fact that there is an increase in DNA methylation, which makes them more susceptible to them. And this is only just the small list. So some of the things that have come out of studying of epigenetics is CRISPR-Cas9. This is an amazing revolutionary piece of science. So CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It is a gene editing tool that basically cuts DNA and then lets natural DNA repair processes occur. There's two parts to this, the Cas9 enzyme and a guide to RNA. So the Cas9 enzyme acts as scissors to cut the DNA at a specific location, which has been identified by that guide RNA. That guide RNA binds to the Cas9, identifies the base sequences of the gRNA, so then that Cas9 will then cut the DNA. There are three categories of gene editing. The first one is disrupting, in which there is a single cut, non-homologous ends join, which results in the addition or deletion of base pairs. This disrupts the DNA's original sequence, which causes gene inactivation. Next is delete, where there are larger sections of DNA that are deleted when they've been identified by two guide RNAs. At this point, they cleavage, and at each site, and non-homologous ends join the separate ends, deleting that base sequence previously identified. And then finally is their correct or insert, which adds a DNA template alongside that RNA that allows the cell to correct and even insert a new gene using that process known as homology-directed repair. So there is another really amazing scientific advancement that has occurred, CRISPR Therapeutics, in which they've been creating gene-based medicines. So they use guide RNA to direct Cas9 to cut DNA to decrease the genetic defect through the process of gene disruption or correction, and then cell therapies, which target certain genes to disrupt to improve the safety or efficiency of therapy. And they may also insert new genes to give cells new functions. There's two types, ex vivo, which is outside of the body, in which cells are removed and CRISPR-Cas9 is introduced to those cells within a culture. And in vivo, in which CRISPR-Cas9 is delivered directly to the person through lipid nanoparticles, which are delivered systematically or to a target. Okay. Some interesting extended learning opportunities include NASA twin study, in which NASA compared the effects of space on identical twins, being able to study the impact of space and zero gravity on DNA and gene expression. There are multiple other case studies that you can explore to focus on a variety of epigenetic topics. Learn Genetics is a great resource for everything genetics. The Bacteria Culture Lab, who doesn't enjoy the hands-on activity, here you can actually look at the effects of temperature have on bacteria growth and their phenotype. Next is a podcast called Big Biology. The episode Genes Don't Do Crap. Dr. Massimo Pogolucci is an evolutionary biologist and philosopher, and he comes on and discusses the connection organisms have between their genes and their environment. And then finally, Queen Bee is a YouTube video in which you can look at bees and the role epigenetics plays in their colony. List of references for those of you who would like to check that out. I hope you enjoyed my presentation and have a wonderful evening.